Abraham, this is group 1C means to say Sarawak 1C, is it? Group Sarawak 1C. Okay. It's okay. You can try to you can you still edit it? If cannot, then I'll just delete it. Yeah. Okay. I'll just delete it. Later you post it again, can? No problem. Yeah. It's sounds legit. Okay, sounds legit. Good. So you can proceed with it, lah. Okay. Now, I think last week or some of your friends was asking me, can we do on some agricultural farm like planting sayur, whereby maybe the fertilizer part you can use fertilizer like cow dung ke, chicken punya feces ke for the fertilizer. Just bear in mind this is animal biotechnology, so the main theme, the main character will still be the animal. Okay, we don't want to complete your proposal and then end up with a plant or agricultural project. Okay, agricultural type of service learning, we try to avoid that. So please make sure we still animal type of project. Okay, now also your friends was asking me, can I go to a food company like a fish ball company, chicken nugget company, or a slaughterhouse, or egg punya a poultry farm? Definitely can. Okay, or even if you know someone who are working on animal feed materials punya factory, also can. Okay, not necessarily you have to see that animal there inside the farm or the company. Okay, as long as it is relevant. Tapi janganlah sampai sayur mau relevant dengan tai ayam, then it's a bit too far lah. And unless you focus on the tai ayam punya fertilizer, where does it come from? From the collection all the way until all the processing, okay, until all the additives that they add to it before they go for the agricultural farm, then probably can. Okay, get what I mean or not? So we try to focus. Now, last time your senior also worked on belacan farm as well. Okay, so they have to visit the farm where not visit, bukan belacan punya farm lah, but the processing punya factory lah. It's a very small scale punya, what would you call it? House kind of kampong type of industry. So basically, when the nelayan take all the fish back from the sea, they will select out the bigger ones. They will sell it at a wet market. The smaller one they are going to bring back, and then they are going to scrap off all the meat, and then they are going to process it to become belacan. Different types of belacan. Bintulu punya belacan, the other type I cannot remember really, but they compare these two type of belacan. Okay, which one is nicer in terms of additive? Which one seems to be better, and which one can last longer? And is there any differences between the processes that were used or the technologies that were used in order to produce this belacan? And you will find that they can find lots of information on it. Okay, especially when they find out whether or not people have been saying eating too much belacan may cause certain type of cancer. Okay, then they try to find out certain publication which to support their work, to tell that what kind of ingredients that were used, which is prone to be converted into carcinogenic materials. When you consume it, it may poses harm to your health. So those they have covered before. And then so you guys can take the ideas and try to come out with something interesting as well. Okay, so that's about it. Remember to post it here. Two group, one single representative. Tak boleh one single representative kan? Yeah lah, can lah. One single representative from the both of the group, and then you write a title here. Okay, unless you guys team up, but you are trying to divert a little bit on the title. Instead of focusing on egg, you want to focus on the broiler, which is the meat chicken, whereby we are harvesting all the muscles, the tissues. Okay, so you want to focus on broiler, then can as well. You have one single representative, and then write out the title, both of it. Okay, both of it over here. Just post it inside this elite link. Okay, at least now, what is the purpose of doing this? So that there is no overlapping. I have previously seen 4 groups doing the same title before as well. Just coincident lah, because everyone is in Sarawak, everyone is in Kuching, and at the end of the day, everyone seems to do the same thing because local punya market, there's not much of a difference. We have CCK, so everyone went to that CCK. So we have 4 groups interviewing the same person. So it is inconvenience for our industry as well. We will try to limit the number, maybe like 10, and then try not to take up too much of the time. Okay. Now the interview process, it can be online or it can be physical. If physical, try to reduce the number that went to the company. Okay. At most, maybe like 3 or 4 of you guys. Janganlah sampai all the 10 of you guys go at one shot. Later there's an outbreak or there's a cluster from animal biotech peer group, then seriously, I'm going to be really famous on that. Okay, I don't want to be famous on this kind of thing. Yeah, okay, so make sure you reduce the number, practice the SOP. Okay, keep yourself safe. If your friends is not really keen into doing physical punya visitation, don't force them. Don't force them. Later post in TikTok ke, whatever ke. Yeah, seriously, again, you are famous. We don't want to get famous these days for the wrong reason. Okay, now any questions so far before we put down the list? Yes. Yeah, can. No, no one paper. The 2 groups, each of you guys have their own respective proposal, your own letter, your own product and execution. Okay, except for because you have your peer assessment. Peer assessment you are going to evaluate each other lah. The rest should be different. Even your student journal should be individual one. Get what I mean or not? Not. Yep. 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 You can. Oh, cannot. Don't copy and paste. Treat it as if they are your collaborators, not your competitors. But yeah, I'm going to, of course, since you guys are doing the same title, there will be indirect comparison of who is doing better. Some people write much better than the other group. Some can execute much more better. Okay, in terms of putting in later, they are going to put in a lot of pictures, the surveys, and tons of stuff. Okay, so I want to do for me, the comparison should be there. Okay, but now, you can see service learning, this is your 70%. The site visit application letter should be 2 because you are collaborating with each other. Group 1C and 1K. So Group 1C will have 1 letter. Group 1K will have 1 letter. Okay. Proposal writing. Now, you can start with your proposal already. You can write like Abraham, since you have your title ready, you write on your proposal. Later I'm going to tell, I think the proposal, I have these different what is the section that you have to mention inside your proposal, right? If you refer to the assignment link that I have in eLib, you're going to see, just click on it. There has to be introduction, research, punya background ke, introduction ke, and then what is the objective, what is the approach, blah blah blah. You try to read on it first. If you don't understand on certain things, you can ask me. Now, proposal writing, remember again, Group 1C will have 1 proposal, Group 1K for another proposal. Okay, execution, same as well. This execution will be much more focused on later on your presentation. So 1C, your own presentation, 1K, your own presentation. Okay, and then how about student journal? This student journal will be individual. Every single one of you has to have their reflection. This is the reflection part, meaning to say, since you have your e-portfolio, right? Your e-portfolio for student journal, you have to have a pre-service learning, meaning to say, before you go for the service learning, what do you feel about this particular sulam? Or you can just go Google, try to find out what is this sulam. Sulam is not like a biotech punya thing or a Dr. Chung punya thing. No, I'm not that. I'm not that into that particular thing. But it is a national approach in Sulam. Sulam is a national service learning whereby they want all the tertiary education punya students to be able to cope with the industry, and they have a social punya contribution towards our industry. Okay, so everyone has to have their own reflection. Okay, has to have a reflection too. You can take your personal picture. Every single one of you likes to take you have your phone, right? You take whenever you go for visit, whenever you do your meeting online, you can take a screenshot and then you post it inside there. When, until when, how many meetings were actually conducted, and who's the leader of the team. Okay, and then that is whatever you guys have discussed, you put it in as well. And what is the point that you have discussed, and then your feelings towards it. You can see, hey, Abraham seems to be a very dominant student. He keeps talking, and I don't have the chance to talk. Of course, I'm not just kidding, like, don't get angry. But yeah, you just put you can say your personal things. It would be your own personal student journal. Not that it is not going to be, it is own will be read by me only. Okay, unless you give the link to to others, your other friends. But of course, your e-portfolio later on will be read by it will be open being space, whereby your potential future recruiter or your bosses later on, your manager, the recruit being manager, are going to read some of this e-portfolio. Don't write too negative things, bad things about your study, and then this particular university is what totally like out of, I don't know lah. There are people who can be really negative. So try to tone down on your writing. Try to be as what do you call it, objective as possible. Okay. Try to evaluate the things based on what you see. Okay. Not too much of your personal feelings. Okay. So you can talk about it. Okay, and then peer assessment later on, it will be group based again. One group will discuss how much marks they are going to give to individual 1, 2, 3, 4, 5 from one group. Okay, so this peer assessment will be group again. So over here, get what I mean? So this is the allocation for all the 70%. So from site visit, application letter until execution of products, your presentation, all in group. Peer assessment also group, but it will be individually evaluated lah, per person. Okay, you guys are going to discuss which person you want to give 4 marks, 10 marks, 8 marks, 7 marks, depends on you. Student journal, individual. Okay, am I clear? Anyone wants to ask any question? Because you guys all seems very confident. No question
Yes,
at all.
sir. I have a
Yes, sir.
question.
Oh, okay. So that, yes, we are very confident.
No,
Okay,
sir,
okay.
I have a question.
Mm-hmm.
Okay, so for my group, right, we have a few choices, but one of it is like a zoo farm in the city. So is it okay for us to like do something about animals and tourism or something? Is that okay?
Can. Definitely can. Animals and tourism. These days, it is I think it is very in punya thing lah. Whereby previously I showed you guys that there's a course briefing slides, right? So just right over here. This right over here. This is VH Green Nature Park. I'm not sure whether you guys have heard of it. It's quite popular in Kuching as well. It's situated in Matang, whereby there are lots of poultry animals, farm animals, so you can bring kids over there. It's kind of like a tourism lah, not really tourism, but yeah, it's a leisure park whereby people go visit, try to enjoy, try to have a escape from the city kind of life. If you are from city, where are you from? Velasini.
Yeah, Villa. From KL.
Oh, you're from KL. So KL,
Farm in
KL.
the city. Yeah.
Oh yeah, can. You can try to make up a team and then try to visit that particular farm. Also okay.
All right. Thank you, sir.
Yep. Any other one? Other people who wants to ask questions? Kita sudah. Oh, you guys are really. Yeah, Abraham. Can. Can. Differences tak apa lah, but you don't share the all totally same writing, the introduction, the research background, the objective. If everything the same, then this is like copy and paste or it's plagiarism really lah. Okay. So if it is just references, yeah. Set references, definitely can. Yeah, because They definitely can. Objective, if overlap, the way you write it will be different, right? The meaning can be the same, but the way you write it is definitely, there's no two person. If I'm going to take you and another, maybe Villa, you guys sit down, I'm going to give you a point. When you put it on paper, it can be totally different. The structure can be different because the interpretation is different. Okay, even though you have a common goal, common object, you have discussed about it before, but when you put it on paper, you will know there's going to be difference. But references, of course, references, you refer to the same materials, you have to be objective enough to put that same materials in your reference list. So that one is okay. As long as you don't tell me point A, point B, even the structure itself, the bahasa semua sama aja. Okay, and then that is bluffing already lah. Get what I mean or not? Okay. Okay. Photo, don't share. Everyone has their own phone. Unless, yeah, because you have only, if possible, try to have representative when you go to visit a farm or a company or a production factory. You guys have either one or two representative for the visit lah. Janganlah you up to the other group. The other groups share with you all the bad materials, all these lousy punya photograph. You have to we didn't do the visit. It's okay lah. We just use that lousy pen. And then when during presentation you see the other groups, wow, all their pictures are so nice. Why? Of course you didn't take your own picture. You didn't take the initiative to visit, ma. So yeah, like you have to rely on other people lah. So if possible, you take your own picture lah. The thing that you think feel nice, yeah, you take your own pictures. It will be nice. Okay, don't over rely on the other group. Yeah, some people, yeah, you know lah. There are people. Okay, now any other questions? Yep, sure. No, you won't. But since you guys are same title, normally I will do indirectly I will do compare. During checking, definitely there will be a comparison lah. This fella, oh, they are writing so nice. And then this other fella, totally similar punya thing. Research all the points, every single point. Only the ayat pun sama. The flow also the same. First paragraph, same point, okay. Second paragraph also the same. Third paragraph also the same. Until the fourth paragraph, oh, I know lah. What is going on already. So you know lah. When your marks come out, you don't lah go. Why our marks are so low? Yeah, you understand. Okay. But then I'm going to post up these rubrics as well. So you follow the rubrics in your own writing. That is very important. Okay. Cool. Okay. Cool. Any other questions? Kalau tidak, we are going to start with our class. Oh, 20 minutes ago talking about this. Have you guys all taken your attendance already?
Yes, doctor.
Yeah.
No, sir.
Okay, faster. I give you 2 minutes or 1 minute. So I want to take your attendance. Then we're going to start.
Doctor,
Yeah.
I have a question.
Mm-hmm.
For the visit, right? May I know like what should we ask or do during the visit?
Okay. Now you have to, before you go for the visit, try to draft out certain questions, maybe 10 or 20 or 30 questions. Okay, each of the question maybe it has to be a progressive one. Don't be a close-ended punya question, right? Or close type of question like yes or no punya question. Try to avoid that. Okay. After you have drafted this 10-20 question, you can show it to me. I'm going to go through it with you. Let's see whether or not these questions are relevant or not. Okay, and these kind of questions, you put it inside your proposal as well. Okay, meaning to say those are your approach. You want to find out whatever information from inside from that particular industry, for that particular company. Then you have to ask certain questions. Okay, how long has this company operate? Okay, what actually triggers the interest into setting up a fishball company, a nugget company. And then you can ask about, since there are so many competitions in the chicken nugget industry, how do you guys actually manage to thrive and sustain yourself for such a long time? Get what I mean? Or there are many, or you can ask another question, maybe like there have been a lot of reports on the harmful effects of pre-processed food consumption. It may cause cancer or may cause blah 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 blah. With example, and then you can add on according to who's research, it has been found that upon eating too much of chicken nugget or fish ball, just as an example, and it has been linked to a increased occurrence of cancer among children, just as example lah. Okay, so you can use this kind of information. Don't be too rough on the question, too blunt as well. Be polite, yet it has to be informative. Get what I mean? If you don't know what I'm talking about, you try to go watch how these BBC people do interview, how these CNA insider people do interview. You ever heard of CNA insider? Don't every day go through your TikTok only. You try to watch YouTube. You watch YouTube, but watch those news channel. How do they do a proper kind of interview? Okay, and then how do these people what do you call it? Turn or maneuver the questions when they are being given a yes or no punya answer. Get what I mean or not? Try to watch the professional, how do they do interview, and then you learn from there. Because every single group over here, you have your own different niche, right? Some poultry farm, some cattle farms, some fertilizer farm. So all totally different. I can't really give you a set of rules, a set of questions. These are the 10 type of questions you have to ask. I can't do that. But you try to go watch this CNA Insider. That many times they will post there's a lot of this documentary lah, whereby they ask questions which are very relevant. Each of the question is going to be connected to each other. Okay, and then they talk. I think there is another documentary on chicken nugget as well. Okay, and then what is inside chicken nugget? How many percent of the chicken nuggets are actually really chicken? How many percent are actually flour? And how many percent of flavorings? How many percent of preservative? And are these preservative beneficial to human body, or are they going to be are they going to cause adverse effects to the human body? Yeah, what I mean? Okay. Are you guys following me?
Yes. So if we go to like two places, then for each farm, like because the farm only have like one type of animal, so two groups we just go to two places. Is that okay?
Two groups go to two places. Why not just one group go to one place, the other group go to another place?
Because
and then you can share your experience together, or you just want to focus on two?
Because we thought like if we go to one farm, then the farm only has one animal. So I thought we have to like at least two animals, right?
What is your title again?
We haven't come up with a title yet.
Sure. You come up with your title first, and then from the title only there is it's like kind of like you do your experiment. You cannot have temperature as your variable, and then you have your what else? Temperature as variable, and then weight or mass as variable, and then I want to use color as variable as well. This you cannot have so many variables. Either you compare farm to farm, maybe cattle in this farm, cattle in that farm, or the chicken processes, the poultry, the boiler processes in one farm, and the egg in the same farm as well. Get what I mean? How do they process these two types of products? Try to limit yourself. Don't don't research on things that you cannot actually handle. This is just service learning. This is a 2 credit hours mere course. It's not worth your time to like drag in every single thing. You want to do cattle, you want to do chicken, goat, what else, sheep, every single thing in that farm. Try to limit yourself, because you will find that a lot of your seniors, after they have gone through when they write their proposal, hey, It's not enough lah. 10 pages, how can we fit everything into 10 pages? Or how can we fit all our findings into 20 slides only? It's basically impossible. Then they have to start to cut off here and there. You don't get to present whatever your findings. It may be very interesting, but you don't get to present. Remember, each group are only given or allocated 15 minutes for your final presentation. Okay, bear in mind. 15 minutes only. 15 minutes, 15 minutes only. More than that, I'm going to cut marks off already because there, you know, some students they go like on and on and on and on without focusing or focus on that findings they have. Okay. Any other questions? No more? Okay, then we are going to start with our class. Now, last week we have talked about. Seriously, no question. Got any question? Just leave it open my slide. Okay, we are going into lecture 2. Okay, now last week we have talked.
Sorry, doctor.
Yeah.
Sorry. Doctor, may I know how do we choose a suitable title for the service learning? I mean, what is the requirement that for us to choose the criteria for us to choose the title?
Actually, it's quite free as long as it is animal relevant. Animal relevant, it can be a farm, like your friend says, Villa says it can be an animal zoo or just simple ecotourism kind of farm, also possible. If you want to go for poultry farm, also okay, or a small scale keropok Lego making kind of in-house or house kind of family-based kind of business also can. Okay, you can try to talk about the process. How do people make keropok Lego from fish? What sort of different additive were actually used? Were there any technological equipment or equipment used in the processing part? Or in small scale industry, they basically use a blender only. You can also talk about it. Okay, and if you were given a chance, do you think you can improve on these kind of processes or not? Okay, by using blender, yeah, you can use blender. Blender like how many times of blender you can blend, okay, before the blender breaks down. But I think last week or when I actually see there is keropok Lego in Kelantan ke atau Terengganu, whereby they have this serious huge blender machine. And whenever they blend, I think it's more than 100 or 200 kilo. Okay, that is a small, although it is called a small scale industry, but as long as there is like more than 20 or 30 what do you call it, pekerja kat sana, okay, it is considered as SME already. Okay, you can actually talk about it. You can use that kind of title. Get what I mean or not? So it's really free. There's no limit to it. Remember, this particular course is really about initiative and the creativity that you put in. Okay, it is not like the usual kind of courses that you take, whereby you go for your final exam, you memorize your slides, and then you finish your assignment, submit your assignment. Ta-da! That's about it. You get your 2 credit hours. There's no assignment, not much of assignment, not much of quizzes, not much of a quizzes, several quizzes lah. And then forum, there's no final exam for you as well. Okay, most of it just freestyle. You know what I mean? If you have posted, if you are really not confident about the title that you're working on, you post it in the link just now. Once you have posted it, you tell me so. Ask me to check on it. Get what I mean? But remember, I have sent you guys my schedule, right? You don't go like at night time. Doctor, can you check my title? I posted in Elite 9 o'clock or 10 o'clock in the evening. How am I going to check for you? I'm not that hardworking every day sitting in front of my desktop lah. Get what I mean? So around office time, you just text me, Doctor, I have posted this and this, or you just directly send me the title. Is it suitable for my group to work on it? I can give advice on it. Okay, cool. Okay, fine. Yeah, everything okay, right? Okay, last week we okay.
Doctor.
Yep, sure. Okay, last week we talked about this CRISPR-Cas genome editing. genome editing. Okay, and then before that, we just basically go through what is cloning, what is plasmid, what are vectors. Okay, and then when we talk about vectors, there are different types of in natural occurring vectors. We have plasmid, we have bacteriophages as well. Bacteriophages, whereby these viruses infects bacteria, and then we can use this type of natural occurring vectors and then bioengineering in order for us to do cloning. Okay, now there are also other types of vectors as well, whereby we can have cosmic, whereby you can take out this cos site. Okay, if you do read on it, there are cos site in bacteriophages genome. Okay, you take out this cos site, fuse it into a plasmid, then it becomes a cosmid. Okay, it is an artificial structure in order for it to perform like a vector. And then what else do we have? We have another one. We have artificial chromosomes. Okay, last week we talked about artificial chromosomes. What sort of artificial? What are artificial chromosomes? Right now, in this case, we are still using artificial chromosomes. Okay, for cloning purposes. And then these cloning purposes, we use it to create libraries. Okay, if we want to store up chunks or the genomes of a mammoth, of a extincted animals, extinct animals. Okay, we want to store the library of it. We have to put it into artificial chromosomes. Okay, artificial chromosome allows a huge inserts up to kilobases or tens to 1,000 kilobases. Okay, to be inserted into these artificial chromosome. Now, where does these artificial chromosomes end up in? Inside hosts, so that these hosts, mostly bacteria, can be stored inside freezer later on. Okay, whenever you heard people talking about the gene what do you call it, germplasm bank, whereby you say, hey, this particular animal has extinct already, so we want to store the genome. How do they actually store it? They don't store it as free-ended DNA or DNA which are you just chop it into ice and then put it into freezer. No, we don't do that. Okay, because it is not going to be long lasting. Your DNA tends to degrade under unprotected condition. So how do we actually protect this kind of free DNA from degrading? We have to put it into vectors. Okay, either circular ones or artificial chromosome, whereby both the terminal end of this artificial chromosome are protected by telomeres. Still remember or not? So this artificial chromosome, then we put it into either yeast, bacteria, and then we create a plate called the bank. Okay, the genome bank, the germplasm bank, and then we store it inside freezer. Okay, so that later on, when we do have the technology already, we can actually use these sequences, reassemble that particular animal back. Get what I mean or not? It is possible. Okay, and people are doing it in mammoth. People are doing it in some sort of extinct cattle. Okay, there are a lot of efforts going on in different corners of the world. Okay, just have to read on it. Now, over here, and then we go further after we have understood about all this cloning system. We want to go into genome editing. Okay, last week we talked about began to talk about what is genome editing. Genome editing is unlike your gene cloning, whereby you put in a gene and that particular cell you want to multiply it so that you get multiple copies of the same nucleic acid. Over here, this genome editing, basically we want to edit the genome of a living organism so that later on it can change in its trait. Okay, transmissible trait or inherited traits. Okay, want to modify it. So the modification itself can use this type of tools. There are 3 types of tools that last week we talked about. You want to consider homing and endonucleases as one of the two, but it is quite simple. Just nucleases binding to DNA, which cuts it, and then from there you get your modification. Or we can have these zinc finger nucleases. Again, what is zinc finger nucleases? Zinc finger nucleases are basically nucleases which are fused to zinc finger binding domain. Okay, it's DNA binding domain, zinc finger DNA binding domain. So we have this fourth one as the nuclease. Okay, zinc finger binding domain as the one as the protein or the domain which binds to DNA. Okay, now again, what sort of things you just put inside, you focus first, what sort of things can actually read DNA? Again, I repeat, what sort of macromolecules can identify and read DNA? Carbohydrate cannot, okay, lipid cannot, protein can, and nucleic acid also can. Again, I repeat, your carbohydrate can read DNA or not as more macromolecules. You know your four macromolecules and your carbohydrate, your lipid, your protein, and then your nucleic acid. Okay, which of these can actually bind, can recognize DNA? Okay, protein, and what else we have? We have nucleic acid.
Nucleic acid.
Yes. So over here, two types, ZFN and TALEN. These are protein-based recognition, whereby this 1F, 2F, 3F, 4F over here, these are zinc finger DNA binding domain. Don't look at this DNA binding domain thinking that they are DNA. They are not DNA. This 1F, 2F, 3F, 4F, they are actually protein. Okay, protein which is capable of recognizing DNA, and then yeah, they are capable of reading it. Either they transcribe it or they do DNA synthesis on it. Okay. Now, last week we've mentioned that zinc finger binding domain occurs naturally inside eukaryotic punya cells, inside eukaryotic punya gene. Okay. In human, there's around 1004 or 1005 genes which has this zinc DNA binding domain. Presence inside the gene, whereby it can be recognized by this zinc finger DNA binding domain of the protein. So we can actually use this zinc finger DNA binding domain, fuse it with nucleases, so that later on we can target these 1004, 1005 genes. Okay, we can either do modification on it. So that is one approach. The other approach, we call it as TALEN. This particular TALEN, transcription activator like effector DNA binding domain. This is the colorful one. Okay. Now, why is these two different? These two are different in that this is isolated from, I think, what is the species already? Yeah, just a minute. Okay, now it is written over here already. Okay. So it is isolated from bacteria. TALEN is isolated from bacteria, Xanthomonas species. Okay. While ZFN is usually happening in all types of eukaryotes. Okay, eukaryotic cells. How about CRISPR? Originally, it is found in this bacteria, Streptococcus pyogenes. Okay. So now, we have these three main kind of approach in genome editing. Okay. Now in Cas, what is this? How is this Cas being used? Two main components of Cas, this CRISPR-Cas or CRISPR technology is first the Cas protein. That is the Cas9 or Cas whatever, like Cas12. So we have the Cas protein. This Cas protein function as what do you call it? As the nuclease. Okay, how about this? gDNA, or we call it guide RNA, no, not gDNA, gRNA, guide RNA. This guide RNA becomes the recognition site. So instead of using protein to recognize nucleic acid, this gRNA or the CRISPR technology uses nucleic acid to recognize the sequence, the sequence that it wants to cut. Get what I mean or not? So over here, PAM is the anchoring part for your Cas9 protein. It will anchor and it is going to cut at a nearby site. Okay. Now, so CRISPR-Cas genome editing principles. Where does this actually happen? Just now we mentioned about it is being found in bacteria Streptococcus. Streptococcus, whatever. Okay, so over here, what happens is that now in this bacteria itself, this is used as its what do you call it? It's immune kind of it's analogous to the human being immune system. whereby throughout evolution, bacteria or E. coli are constantly being being attacked by bacteriophage, constantly attacked. Okay, so what people this Doudna found, okay, the researcher that found this CRISPR-Cas protein is that she found that inside inside just a minute inside this bacteria they actually stored chunks and chunks of bacteriophage punya RNA. They'll cut it and store it. Cut it and store it inside the genome. Then we'll probably be asking, what's the purpose of this bacteria? What is he trying to do? Why trying to take chunks and chunks of this nucleic acid and put it into their own genome? Well, they are trying to put it inside them so that they have a memory of what they have survived through. Okay, if this particular virus have actually entered their cells and then didn't cause them to die off, they will take chunks of these viral punya sequences, put it into their own genome. Okay, right next to this Cas punya protein, Cas9 punya protein, Cas punya cluster protein. This Cas punya protein, every time, whenever there's a particular invasion from the bacteriophage or from invasion of other virus punya DNA or nucleic acid, they are going to generate their memory. Okay, try to revive their memory. Has this virus in evolution, in history, have they actually affected us before or not? If yes, they'll quickly synthesize this CRISPR RNA. Okay, CRISPR RNA, they are going to this RNA. Okay, they are going to synthesize it. Okay, fuse it together with the Cas protein and immediately attack whatever invasion they are actually experiencing. It's kind of like your T cells or B cells, whereby they have the memory of the antigen which have previously affected human. So they store this memory, and then whenever any viral genome or viral nucleic acid come in, they can immediately recognize it, cut it, okay, disarm it, so that they won't be affected so much by this particular viral invasion. Okay, what I mean or not? So over here, so what we did was that to take the whole Cas cluster, CRISPR cluster, Cas protein cluster, whereby normally it is going to be, there's a CR, there has to be a Cas, yes, the Cas gene is over here. This Cas gene fused together with all these CR, tracer RNA. Okay, so basically this tracer RNA is the one we try. If we are going to hijack or manipulate this particular system, we can put in the DNA that we want to target, or whatever sequences that we want to target in the species that we want to modify or edit, genome edit. We put in the sequence, fuse it together with this cRNA, okay, so that it can become our gRNA, single guided RNA, or we can gRNA or single guided RNA. Now, this single the guided RNA, when we actually introduce it into the whole cell, what it does is that whole cells over here means to say the species that we try to modify or try to edit. So what we do, the Cas will be expressed, and this Cas protein is going to take this, express this guide RNA, fuse it together, okay, and then it is going to go into your cell, okay, and then it starts to recognize which area to edit. How does it actually recognize? Using this tracer RNA, okay, that we have modified and put in. So it will go search all the way inside your genome. Hey, this is the location that it binds already. So they are going to bind, recognize the sequence inside it, and then start to chop using this Cas protein. Okay, now then what is the outcome of this chopping action? Okay, what is the action? What is the outcome of it? We are going to talk about it now. There is 3 different outcomes: SDN1, SDN2, SDN3. Now, why do we have to have this SDN1, SDN2, SDN3? Because this is how right now currently we try to recognize what sort of editing or the outcome of your what is the outcome of your genome editing? Is it what kind of changes actually appear, the physical changes, the structural changes that occur inside a genome. In SDN1, what actually happens? There is a deletion. It experiences once the CRISPR-Cas protein cut it, it experiences a loss or a few nucleotides, a loss of it, deletion, okay, loss of it. And then during the repair process, DNA are being joined back again, and then it loses one or several nucleotides, okay, without introducing anything at all, okay, the whole process, but loses one of two nucleotides. Now, in the second scenario, SDN2, or we call it site-directed nucleases, in the second scenario, what happens is that change with templates. Certain nucleotides were introduced. Maybe maybe 1, 2, 3, 4, 20, up to 200. Okay, up to 200, the standard. That normally people take this 200 as the limit. If it is more, they consider it as SDN3 already. Okay, this is small changes. These are small changes in the genome. Okay, we insert it. Okay, which causes a change in characters, change in the traits of the animals that we want to edit. Okay, SDN1 the same, SDN2 the same, SDN3 also the same. SDN3 is whereby a huge chunk, okay, whereby during the whole process, we break off using CRISPR-Cas or using ZFN or using talent. It's broken and huge chunk, maybe a few hundreds, all the way until few thousand kilobases were a few thousand bases were actually inserted into the genome. Okay, get what I mean or not? Now, other than these three scenarios, we have 1, 2, 3. There are also other scenarios whereby we call it as what do you call it? Cisgenesis or cisgenesis, not transgenesis, intragenesis. What is cisgenesis? In cisgenesis cases, we are taking a stretch of DNA from a similar species of animal and put it into another individual, in another population. For example, over here, we know Mat Saleh lah kan. They are tall. Sometimes they are huge. They have more muscles than the Eastern punya people. So we can take the population, the DNA from them. We are still all considered as Homo sapiens, human. But there are certain genes in the Eastern kind of Asian kind of population whereby, of course, if this is just an example, we can take the gene from the different population, although we are same species, take it and put it into the Asian population. And then we can grow like huge, taller, stuff like that. An example, okay? Or you can take maybe a cattle population, the lembu population from Himalaya region, and then from Indonesia, you say, hey, these cattle in Himalaya, they are capable of surviving extreme climate during a very cold season. They are not affected at all. Okay, and then they can live with a minimal amount of what do you call it? These feeding materials, very minimal. So is it possible that we can transfer certain genes from this population, although they are both the cattle species, but in Indonesia species, we don't have that particular gene. Can we take the gene and then put it into the Indonesian cattle? Now, this particular scenario is what we call it as cisgenesis. Cisgenesis. Okay. There is also another scenario. We call it as intragenesis. What is intragenesis? Intragenesis is whereby you take a gene from another species and put it in another species. Intragenesis. That is the outcome. So we have SDN1, SDN2, SDN3, cisgenesis, intragenesis, and then there's another what is that? ODM or directed mutagenesis. There is a mutagenesis as well, whereby oligonucleotide directed mutagenesis, ODM. If you have pen, you want to jot down, but it is not going to come out in your exam. This is just for your extra knowledge. Okay, so during the genome editing process or the gene modification process, we have different scenarios. Okay, it can be a slight change, a single nucleotide change, or a deletion, whereby is SDN1 totally not detected, SDN2 few nucleotide changes, okay, where it quite least to be detected as well, okay, not really that significant. In SDN3 is those that you will normally talk about genome edit, what we call GMO, whereby huge chunk were put in. Okay, and then we have oligo-directed mutagenesis, whereby instead of changing anything inside the genome, we just want to change one single nucleotide or two nucleotide. Okay, and it is used, we are using oligonucleotide site-directed mutagenesis. I believe you guys heard about it somewhere along the way. Okay, so oligonucleotide mutagenesis, whereby we use a primer bind to the sequence that we want to recognize, and then using other proteins, it is going to change maybe A adenine into a thymine, or cytosine into a guanine. Get what I mean or not? So oligonucleotide directed mutagenesis, mutate it, okay? Or it can be cisgenesis or intragenesis. There are plenty of other definitions going through. So over here, you can see knockout. Okay, so gene editing detection in biosecurity, we can use for NHEJ repair DSB, provide a percentage of alleles that have been edited with a mixed cell population. Now, just remember the approach. Okay, we can use mismatch cleavage assay. I'm not going to go into the detail how does it actually happen. Okay, and then we can use restriction digestion. This we are going to cover later on in genetic markers. I don't think we have time. Okay, and then PCR amplification. You can use PCR. Again, we are going to cover in your genetic markers later. And then we can use sequencing as well. Basically, you sequence everything. Try to find out whether or not there has been changes inside the nucleotide. Okay, these are the different approach for identification or gene editing detection. Remember this one. Why is it so important? Because Malaysia, we have biosecurity council as well. Meaning to say, whenever you want to do a gene editing or genome editing or genetic modified organism, you want to create it, we have to submit a proposal or form. Okay, we call it the Annex E form. We have to submit it to this Biosecurity Council of Malaysia. Not that they want to control you or anything, but they just want to keep you in check to see whether or not you have the required system or environment in order for you to conduct such a research. Okay, just not that it is because our this genetic modification is definitely going to be detrimental to the environment. Not really. Just because it is so beneficial, there's a lot of advantage to it. Okay, that's why we want to control it, so that when things are going on, there is a guideline for you guys to follow. Okay, don't think about genetic modified organism as something evil, or it should be totally avoided. As a biotechnology students, we want to do more of this research. Why? Because we want to improve the yield crop. We want to improve the yield that comes from our agricultural products. We want to fit the overall growing population. Okay, without much harms to the environment. Okay, without doing much harm to the environment. So that's why there is a biosecurity law in Malaysia. Okay, biosecurity. I think this biosecurity law will be covered by in your third year. Okay, whereby Dr. Alexson will talk about all the latest updates on these biosecurity guidelines and laws. Okay, you will listen to it later on. Okay, so right now you just learn about the technology. Okay, so SDN1, okay, differences in gene editing detection. The detection itself, okay, how for SDN1 is very difficult for you to distinguish, indistinguishable compared to natural mutation, trait selected through breeding, because we are only talking about a loss of one or two nucleotides only. And the loss of this nucleotide, although it is just a loss of one single nucleotide, it may cause a frame shift inside your gene coding region. You know what I mean or not? Meaning to say that particular protein cannot be used anymore. Okay, because we know last week we talked about it. Every 3 nucleotide contributes to 1 single codon. Okay, 1 single codon will tell you what kind of amino acid will be incorporated into a protein. Okay, now what happens if from these 3 nucleotide punya codon, I take out 1 single nucleotide? A frame shift will occur, meaning to say the subsequent nucleotides, it is not going to be read 3, 3, 3, 3 anymore. The frame will be lost. Okay, and the subsequent codon will be read differently, and keep going, and the whole protein will be a junk. It doesn't make sense. It doesn't work anymore. Okay, so the outcome can be great, whereby it can changes a trait through just 1 single deletion of this nucleotide. But in STN1, it is very, very difficult for a biosecurity officer to detect. As if I'm telling you how to cheat in your exam macam tu lah. Okay, so STN1 is a very good technique whereby you change 1 single nucleotide, but you don't get it can go unnoticed because it is kind of similar to natural mutation. 1 single mutation can lose everything already. Okay, you can lose that particular trait or that particular protein can be gone already. Okay, so SDN1 is very difficult to distinguish, very difficult to detect. Okay, SDN2 detection varies on the size of edit, 10 to hundreds of bases. Okay, it can go up to 200 bases, and type of edit. Sometimes it can be just a snip or a very small edit. Okay, because gene are normally like at least a few kb lah. There's seldom there's like 100 or 200 punya protein punya size, 100 or 200 punya gene punya size. Very uncommon. Okay, so and anyway, SDN3 is easiest to distinguish because large chunk of DNA has been inserted into the host. Okay, so this can be easily identified. We have to have a wild type punya animal with your genetic modified punya animal, put it on a gel, run it, and then this particular fragment will appear. Because you will see it later on, we are going to talk about genetic markers. How do you distinguish between changed and non-changed punya animal? Okay, now these are some of the examples. Okay, we've been talking so much about SDN1, SDN2, SDN3. What are the examples? What kind of animals has been created based on this kind of approach? These are those that have been reported. Of course, there are certain animal modification that goes unnoticed. People have been eating it, people have been breeding it, people have been raising these kind of modifications, livestock up, but it is not reported. Bear in mind the report over here, these examples listed here are those very honest punya researcher, whereby after they have conducted the modification, they actually publish paper, verify it, and publish paper. And then we can list it right here. So what type of crop plants that have been modified using SDN1? Grapes with less branching. So previously grapes are lots of branching, so these days it is being reduced. And then mildew resistance barley overseas as well. Increase nutritional value in rice. Okay, in rice as well. In rice is SDN2. Okay, and then improve grains yield under drought stress, SDN3. Okay, livestock modification about animals. Okay, you can create allergen-free milk from cattle is SDN1, whereby 1 single nucleotide mutation, that particular protein which causes allergen, which causes allergy in you, okay, which becomes allergen, that particular protein can be destroyed. Okay, because 1 mutation, 1 single nucleotide of deletion, you delete it, this 1 single nucleotide, the whole frame shift, the whole frame are being shifted, and probably it pre-terminated the transcription process. That's why it becomes allergen-free milk. Okay, and then with disease resistance animal, also SDN1, maybe in this one CD163 gene, okay, which most probably it is involved in your what do you call it, most probably is involved in the immune system of the cattle. Okay, you delete it off, basically it gets less it becomes more resistant to certain diseases. Hornless dairy cattle, SDN2, cold tolerant livestock, SDN3. Okay, and there are more publication over here. You can see these are all different. We have cattle. Still remember myostatin? Previously, we mentioned that it is involved in the meat punya stress. Okay, so by the knockout of it, it actually increases muscle yield. It also happens in goat, myostatin, pigs, myostatin knockout, sheep, myostatin knockout. So in all these species, because we want them to grow more muscle tissue so that we can have more meat to consume, all these are potential. These are the meat animals that we have, and all of them have been not necessarily the market that were consuming it. All are more genetic modified, tak lah. But this has been conducted before, and it has the potential of being transferred. The technology being transferred over here. Okay, now last slide already. Okay, so this is the process. Although it sounds very simple, like you gene modify satu nucleotide, oh, we get hornless cattle, we get disease resistant cattle. Sounds very nice, but the whole process takes years, okay, if not months, years. It basically takes years. So what happens is that first of all, you have to cross over cattle. Cross over, you derive the embryos. The embryos meaning to say the eggs has been fertilized before. Okay, and then you have this embryo transfer it, collect the fetus, establish fibroblast cell lines. Fibroblast cell lines are basically like embryo cell line, kind of like a very stem cell line, a primitive cell line, we call it. Okay, genotype each line, and then select line with best genetic merit. Select it out, this one. Okay, from there, you start to do genome editing. Okay, edit it, the gene that you want to edit. Okay, once you have edit already, now in this particular process, this is the process that we are talking about. We have SDN1, SDN2, or using CRISPR, or using ZFIN, or using talent. Okay, genome editing, we can do it right here. This is the one only step, the small step. Okay, once you have conducted this already, the next one will be to verify the changes. Send it for sequencing, send it for mismatch cleavage just now, and then or send it for PCR, or send it for what else? Whatever lah. Okay, you're using your nucleic restriction enzyme to cleave it. Okay, but the most reliable method will be sequencing. You just sequence it to see whether or not that particular sequence that you are targeting has been modified. Okay, once it is done, cultured selected fibroblast. Okay, culture it. Okay, and then we introduce this fibroblast into this is called somatic cell nuclear transfer. Put it into a empty punya egg, the egg that has been enucleated. Okay, this egg over here has dia punya genetic, dia punya DNA semua dah kena buang. Okay, kau masukkan DNA yang dah modify tu, combine dengan dia, and then you put it back into the animal, the mom just now. Okay, and then you carry it for 9 months or 10 months or a year before the genetic modified punya or genetic edited punya calf being born with the trait that you desire. Okay, so the whole process is complicated. Okay, it's complicated. It has to start with crossing it, deriving the embryos, collecting the fetuses. From the fetuses, you collect a potential fibroblast. From the fibroblast, you genetic modify, genetic edit it. You have to confirm. After confirm, introduce back into donor cells using SCNT, stem cells, nuclear transfer. Okay, back into the enucleated cells, into the parents. Let the calf grow, and then from the calf, after 9 months of carriage, it is being delivered. Then you only then you have your genome edited calf. Okay, get what I mean or not? Now, last week your friends shared this last week, or was it last week or a few days ago, your friends shared this BBC punya news kan? What is it? Yeah, Shafiqah atau siapa? Who shared about this pig punya kidney being transplanted into human. Correct or not? Okay, now the technology used, CRISPR as well. Okay, whereby they try. Now, previously, of course, we know in conventional punya medical settings, we have to wait for a donor. And a donor is really hard to come by unless you are taking from your close relative, from your parents. So in the news itself, if you go read it, you will notice that the mom or the family members actually are willing to donate their kidney to that particular brain dead punya individual, their family members lah. He's brain dead already. Okay, tak boleh it cannot be healed already. Okay, just waiting for his time. But in order to prolong his lifespan, they have been thinking whether or not to take their own kidney and donate it to that brain dead individual. Okay, but then these doctors approach him, the researcher approach the family members to ask whether or not they get the permission to transplant, to genetic modify pig's punya kidney. Okay, genetic modified it so that it doesn't causes much of a immune stress. Dia tidak kena apa, what do you call it, rejection. They try to immune rejection, they try to overcome the immune rejection. So what they did some modification, okay, using the same method, genome edited, okay, on the immunity cells, okay, immune punya gene. Once they have edited already, transplanted it onto that brain dead punya patient. Okay, and they notice that the kidney starts to function as usual. So then people will be asking, hey, since we are Malaysia, is it possible that we use cattle punya ke, kidney ke, or other types of animal, maybe chimpanzee ke? For your information, pigs, why do scientists want to work with pig? Because the size of the adult punya pig, the size, the structure of the organ is very similar to human. Dia punya size dia. Okay, for cattle, of course, you are expecting the physiology and the size of the organ is much more larger than human. Okay, which becomes a misfit. And then another thing, probably you will ask, why not we just take from primates? Most of the primates, they have these zoonotic diseases. Okay, zoonotic diseases are very dangerous. You don't want just like this. This time we have this COVID punya cases, also a zoonotic disease, previously a zoonotic disease, and then it is being transferred when people consume bats. Okay, then it is being transferred to humans. So zoonotic diseases can cause a havoc to humankind. Not that we don't want to try it, but from primates, the zoonotic diseases are very dangerous. Okay, try to avoid it. So the most stable is pigs because it has been domesticated for centuries. Okay, and then due to their domestication, they have been living with in farms, a controlled environment, so we can basically predict what sort of zoonotic diseases occurs in this pig. Okay, and also from the article itself, I think it has been mentioned that these scientists have genome edited some of the genes present in this pig so that they don't transfer the usual type of zoonotic disease from pigs to humans. Okay, so a lot of medical breakthrough has to do with genome editing as well. Okay, genome editing. I think that's about it for these slides. Let's begin with our second part of the class. Okay, you guys, you want to take a break? You guys are taking break already. Hello, are you guys there?
Hello, Doctor.
Yes, doctor.
Are you guys following what I'm talking about? Oh, I'm shocked sendiri saja.
Yes, Doctor.
It's like monkeys
About
already.
pig.
About pigs. Okay, fine. So yeah. Should I just continue?
Sure.
5-minute breaks. Can. Yeah, 5-minute breaks. And then come back again. Mhm. Yep. No worries on. This is combined class, so no worries. Just take the attendance. Later on, yeah lah. No worries. I'm not going to bar you from exam because this later on, if your what do you call it, your academic advisor asks, because your academic advisor punya system, you can actually trace whether or not you come to class or not. So all this later if they ask, you say it is a combined class now. Don't put it in. So your attendance was taken from group 1 instead. Okay. Ask them to approve it because you need your exam slip later on. But yeah, we don't have for this course we don't have final exam, so no worries on it. Yeah. Okay. Now you guys again, I'm just going to start with new class already. If you have any question, stop me. Okay. Don't forget to ask. I know all this stuff are very confusing unless you have studied it before. Okay. Have you guys studied heard about any genome editing before this class?
Not really, actually.
Not really, huh? Okay. So these are new stuff. Oh, you have heard about it from forums, or what? Okay, nice. So at least this is not too, what do you call it, too novel for you, or this is totally stranger for you, not right? Okay, fine. Okay, now we are going to this before we go into genetic markers. Okay, we have to know some of the DNA manipulative enzymes. What are the different types of enzymes that we can use to change DNA, to modify your phosphodiester bond, to modify your nitrogenous bases, to modify the phosphate group, to modify this phosphate group. Yeah, our DNA are made of nucleotides, and in basic nucleotides, we have three common scaffold, three common chemicals group, okay, or molecules. First is nitrogenous bases, your ACTG, okay, it can be a pyridine or purine, and then what else we have? We have the pentose sugar, and then what else we have? We have a phosphate group. Phosphate group and pentose sugar, they make up the backbone of your DNA. Okay, nitrogenous bases are the protruding one which interact with an anti-parallel strand because DNA are anti-parallel. This anti-parallel allows the reading from 5' to 3' and from 3' to 5' on the other strand. Okay, DNA are always synthesized from 5' to 3'. Okay, and the sequence the template are being read from 3' to 5'. Okay, you have to remember these are the basic DNA structure that you have to remember. Before we can know how to manipulate our DNA, we have to understand the basic structure. Kalau basic structure pun tidak pasti sudah, then it will be very difficult for us to discuss about how to manipulate enzyme. Okay, so all together we have 4 broad classes. Broad classes as very, very general. Okay, there are other enzymes as well, but we are not going to talk about it. Or we classify them under modifying enzymes. Okay, 3 classes over here. First classes will be nucleases. What is nucleases? Nucleases are any sort of enzyme which is able to cut, shorten, or degrade a nucleic acid. Then we call it nucleases. Even nucleases have different classes of nucleases. With exonucleases, with endonucleases. The endonucleases are 1 type 1, type 2, type 3. Different types of nucleases. And then we have ligases. What is ligases? Ligases are basically nucleic enzymes that are able to join DNA together. Okay, you are going to encounter ligases, nucleases, very common. As long as you stay in biotechnology, you are going to encounter this type of enzyme. You are going to use it at any time. Okay, and then we have polymerases. What are polymerases? Polymerases are enzymes which makes copies of DNA. Okay, you can have DNA polymerases. You can have RNA polymerases. Okay, and then they have different characters as well. Okay, so the most important features of polymerases, they make copies. Okay, but of course, polymerases, when they want to make copies, they have to have template. Macam photocopy machine, you want to photocopy something, you have to have a template or a piece of paper that you want to make copies of. Without this particular paper, what are you? There's nothing going to turn out. Copies is just going to be empty copies of it. So in order for polymerases to work, you have to have a template for it. That is what we call template. What is modifying enzyme? There are certain enzymes which modifies by removing chemical groups from your DNA. Like what I said just now, you have nitrogenous base inside your DNA, a simple, the most basic nucleotide. We have nitrogenous base, we have phosphate group, and then we have pentose sugar. Now, this kind of modifying enzyme belongs to a large group. It can be kinases. What is kinases? Kinases are protein which tends to remove or add this phosphate group to your DNA. Okay, if you are talking about, like for example, you have 5 prime, you have 3 prime, right? How do you define your DNA as 5 prime? How do you define your DNA as 3 prime? 5 prime over here means to say your DNA punya backbone, because you have phosphate group and your pentose sugar, right? Pentose sugar, how many what is the ring size? It is a 5-membered ring, correct or not? Your pentose sugar in your DNA structure, it is a 5-membered ring. Okay, the position number 5, it is occupied by what? The position number 5 is occupied by a phosphate group. The position number 3, it is occupied by what? By a OH group. Phosphate, OH, phosphate, OH. So when they combine together, they become phosphodiester bond. Get what I mean or not? Okay, now any enzymes that is capable of modifying the phosphate or the OH group or the pentose sugar, we call them the modifying enzyme. You know what I mean? So it belongs to large classes of enzymes. Okay. Now let's look into dive deeper into nucleases. Anyone recording this lecture or not? Okay, fine. Yeah, at least you you just put it there, and I think Microsoft, they allows us to download on our own. I'll see later. Okay, so that later I can just, if some of you guys have missed the class, at least I don't have to repeat basically. Okay, so now look, nucleases. Now 2 types of nucleases. What sort of nucleases? Exonucleases and then endonucleases. What is exonucleases? Whatever these exonucleases, they would cut from either 3' end or the 5' end. 3', 5 prime, 3 prime, 5 prime, 3 prime. Remember that or not? Anti-parallel. Okay, so over here you have 5 prime, 3 prime. Over here also have 5 prime, 3 prime. But this exonucleases doesn't care. Those free end punya nucleotide, which is not protected by anything, it is going to chop it off one by one until the DNA gets degraded. Now, these are the type of nucleases that scientists worries the most, because whenever you have a PCR product, you have a free end DNA. Free end maksudnya at the very end of it, there's no protection at all. You store it inside water or store it inside buffer. Slowly, these exonucleases were going to chop it off. The DNA is going to be degraded. That's why very often when you go to the lab, people are going to talk about, hey, DNA degraded already, DNA degraded. It causes a smear to be formed on your gel, or your gel, on your gel. Okay, it causes a smear to be formed. Now, why is there a smear? Because of these exonucleases. Their interaction is not specific to the sequence. As long as there is a free terminal end, they cut it. Okay. How about endonucleases? Endonucleases are much more specific. They can cut right in the middle. So probably you'll be thinking, hey, just now the CRISPR-Cas, the ZFN, and TALEN, they use what? FOC1 and Cas9, right? FOC1 in TALEN and ZFN. Okay, so over here it is the same thing. These endonucleases, this particular protein, they cleave right in the middle of a DNA sequence. That's what we call as endonucleases. But endonucleases, very special. They have several different types of it. Okay, I'm just going to talk right here. Now, restriction endonucleases are here. 3 major classes of restriction endonucleases are recognized, each distinguished by slightly different mode of action. Type 1, cut at a site that differs and is a random distance away from the recognition site, at least 1,000 base pair. Meaning to say, these type 1 endonucleases, when they recognize a sequence, they don't cut the recognized punya sequence. They cut somewhere else. Okay, because these endonucleases, they are huge protein, or they can actually pull that particular DNA from a distance, 1,000 base pair away, and then cut it on another site. Okay, this we call as type 1. Due to this kind of characters, or it cuts at random site, we scientists seldom use them as a tool in bioengineering. But in type 2 endonucleases, these type 2 endonucleases cuts within recognition site. What sort of examples we have? These are the examples that you normally use in your secondary school. You heard about ECOR R1, BAM-H1, Hindi3, SAL3A, TAG1, H3, NOT1, SFI1. These are all what we call as type 2 restriction endonucleases. Okay, they give you either blunt end or sticky end. ECOR R1, it cuts right here. Okay, GAATTC cuts G, and then AATTC it forms a AATT overhang. Get what I mean or not? Okay, so it either creates blunt end or sticky end. All these group into type 2 endonucleases because they cut inside their recognition site. This is a recognition site, they cut within this recognition site. Type 3, okay. Type 3 are enzymes that recognize 2 separate non-palindromic sequences that are inversely oriented and cut DNA about 20 to 30 base pair after a recognition site. Sounds familiar or not? Okay, this sounds more like your FOC1 enzyme just now. Nucleases, FOC1 nucleases, because your TALEN and ZFN, they uses FOC1 as nucleases, right? So whenever these enzymes, they recognize the ZFN, they recognize a site, but they cut out of, they're not cutting within the recognition site. They're cutting somewhere else, maybe 20 to 30 base pair upstream or downstream. Okay, these are what we call as type 3, which is very useful as well because it is being used in cars, it is being used in CRISPR, in ZFN, in talents. Okay. Now, the frequency of recognition site, what is this about? Now, can we actually use this to predict? Okay, now what if we have a sample of human DNA taken from any one of you? Okay, a sample. Now we have what? Human, we have around 3 billion base pair, around 2 point something or 3 billion. Let's just take 3 lah. Okay, 3 billion base pair, 3 billion base pair, and then can we actually predict how many restriction site is it available inside our genome or not? Okay, now probably why do we want to cut human genome suddenly out of nowhere? It is very important because it can be used in fingerprinting. Okay, we are going to talk about fingerprinting or genetic markers later on. Okay, it is very important, a very important concept. So can we actually predict how many recognition sites are there present inside a particular organism genome? Okay, so how do you do it? This GATC, let's say there's an enzyme which recognizes GATC, regardless it is type 2 or type 3 or type 1. Okay, just GATC. Now, what if you are going to put 4? How can we actually recognize it? There is 250, so 4 power by 4. Okay, what is 4 power by 4? Meaning to say, if you have G, A, T, C. Now, this 4 position over here, inside our genome, this position can be, what is the occurrence of G inside your genome? Is G. It is 1 over 4, right? In any sort of position, 1 over 4, correct or not? Any position inside your genome. If you point one single nucleotide, you say, is this a G? What is the chances of you guessing right? it will be 1/4 because there's 4 types of nucleotide. You can't really go anywhere anymore. Okay, so 1/4. Now 1/4, and then this second position also 1/4. Third position 1/4. The fourth position also 1/4. Okay, so if you are going to take 1/4 power by 4, we get 256 nucleotide. Now, what does these numbers over here means? Okay, meaning to say every theoretically or mathematically, the permutation will tell you every single 256 nucleotide you are going to encounter one of this sort of nucleotide sequence, GATC. Get what I mean or not? GATC, you are going to encounter it every single. Okay, this GATC over here, okay, GATC, it goes 1/4 times 1/4 times 1/4 times 1/4. So it's equivalent to 1/256. This means that if you have a genome, this is 3 billion base pair, the occurrence of GATC, after this occurs one time already, after 256 nucleotides away, GATC. After that, 256 base pair away again, GATC. You know what I mean? 256 base pair. Okay, then you would say again, go back to our original question. What is the occurrence of this GATC inside a human genome? 3 billion base pair. So it will be 3 billion 0 0 0 0 0 0 3 0 0. Okay, over it will be 256. What is the calculation? So we have it is 11 million. It's 11. It's approximately 11,718,750. Okay, this is the occurrence. How many times this restriction enzyme can cut our human genome? Get what I mean? Of a 3 billion base pair. You guys get what I mean or not? No sound? Or don't? Yes, it's just probability. Okay, so based on this assumption, the nucleotides are ordered in a random fashion. The 4 different nucleotides are present in equal proportion. In practice, neither this assumption is entirely valid. So it is not entirely correct, but it allows us to do a prediction. Okay. Now, this restriction digestion, I'm not going to talk about it. No worries. Electrophoresis, no. Okay, let's talk about ligases. Ligases are basically enzymes that ligate DNA together. That ligates DNA together. Just a minute, I need to text my friend. Just a minute, because they're waiting for me for lunch. Right? Just a minute. What's that? Okay. Okay, so ligases is another class of enzymes whereby it allows DNA to be sealed together. Okay, meaning to say it is an enzyme that forms phosphodiester bond. Okay, phosphodiester bonds are the bond that bind your nucleotide together, and this enzyme is capable of sealing the different fragments of DNA together based on the phosphate group and the OH group. Okay, so it can form phosphodiester bonds. Okay, linker adapters, I don't want to ask. I don't want to, you can read on your own. It's very straightforward. DNA topoisomerases. This one you read on your own. Okay, and now let's go into genetic markers. Okay, again, first of all, these genetic markers, our concept is how do we differentiate individuals within a population or within a species. For example, what is the difference between Villa and Abraham just now? Okay, how do you, of course we don't, this different gender, right? But if same gender, okay, how are you differentiate one person to the other person. If we don't look at the trait, if I just merely have a drop of blood, okay, from both of you, okay, how am I going to differentiate between two of you? Okay, what is the difference between, we don't try, we take aside the blood group first, because the blood group is also ultimately determined by whatever changes you have inside your DNA. So what is the structure inside your DNA that makes these two individuals different? Okay, we have two types of differences. Okay, first of all, we call it single nucleotide polymorphism. The other one, we call it variant of large DNA segments, VNTR. Okay, variable number of tandem repeats, VNTR, or single nucleotide polymorphism. Meaning to say, over here, polymorphism can occur inside each of our genome. Okay, it can occur in any single places, maybe in Vila at position number 39 or 40. A nucleotide is available. In Abraham, in position 39 or 40, a thymine is available, okay, instead of an A. So that makes your single nucleotide polymorphism. Okay, different changes. Now, they will say, can it just be random mutation? Yes, this mutation, this nucleotide polymorphism actually comes from mutation as well. Normal mutation, okay? It can be just, it can be a point mutation. It's just a point mutation. It can be a nonsense mutation, or it can cause a type of disease. Who knows? Okay, but this type of polymorphism, okay, this type of polymorphism, it has to, how do we account, or how do we tell that this is just a random mutation, or is it a polymorphism? By the rate of occurrence. If this particular mutation is up to more than 1% of the overall overall population, okay, more than 1% of the population has this particular mutation, then we consider it as a polymorphism. Okay, polymorphism can be adapted or can be detected using this kind of technologies. Okay, meaning to say, when you watch CSI, when you watch this what special victim units, you have we can have these different types of DNA. How do scientists actually tell between different DNA samples, or whether or not this suspect is the criminal that they are looking for? Based on this type of differences inside your genome, single nucleotide polymorphism and variable number of tandem repeats. Now, what is single nucleotide polymorphism again? Now, how does it, what is the meaning of it? Okay, now every single individuals, you have your own polymorphism. Okay, and then another type of variant is variant number of tandem repeats, meaning to say there are chunks of DNA inside our genome which has different repetitive unit. Okay, in Isaac, a single allele might have been repeated 5 times. In Bali, is it Villa? And it has been repeated 50 times. Okay, so when I'm going to take both of your DNA and run it on a gel, of course, the one that has more repeats will stay up here, will stay up on top of here. Okay, on top of here rather than down here if it is just 5 repeats. Okay, now last point. Where does all this single nucleotide polymorphism and variable number of tandem repeats come from? A very easy answer. A single nucleotide come from your mutation, but variable number of tandem repeats, it comes from your parents, both your parents, and from your parents, it comes from your grandparent. Okay, remember during what happens during your meiosis is different from mitosis. Mitosis, it produces 2 similar identical daughter cells, but in meiosis, crossing over actually occurs, and this crossing over actually allows a segregation of a chunk of DNA between the 2 homologous chromosomes. Get what I mean or not? It changes the position, and if it is being segregated, that is what makes you and your sibling different. Okay, otherwise, if there's no homologous, there's no crossing over, there's no recombination, you and your sibling would not be different. You are going to be same only. Okay, get what I mean or not? And then so unless you are identical twins, every single siblings of you, every single one of your siblings will have different number of tandem repeats due to this segregation. But always you can actually trace the heredity. Okay, you can trace it. Where's the source coming from? Is it from your mom or is it from your dad? Okay, and then on your mom's side, your paternal side, is it going to come from your grandmother or your grandfather? Okay, and then the maternal side, is it coming from your maternal grandmother or maternal grandfather? You get what I mean or not? So they themselves are, so all these all these tandem repeats can be traced. Okay, so I think we just stop here. Next week, I'm going to continue on with the different techniques to detect these two types of differences inside your genome. Okay, so ultimately, what makes you different as an individual? Okay, why is it so deep? Why everyone claims that every single person's DNA or genome are unique to themselves due to this? Okay, these are the two features that you have to remember. Okay, it causes single nucleotide polymorphism, variant number of tandem repeats. This and this may causes different behavior in your gene coding region, changes in your gene coding region, which may ultimately change your characters or traits. Or it may just be a random single nucleotide polymorphism without any effect at all. Okay, without any effect on the traits itself. Okay, it can be. Okay, so next week we are going to talk about what sort of techniques that we are going to use to define these two types of difference inside our genome. Okay, now you can turn on your mic. I think that's about it for today. Any question? At least you ask me some questions.
Doctor,
Yeah. Yes, I'm here.
doctor,
Can you
I
hear me?
don't. Okay. I don't quite get type. Yes, I can hear you. I don't quite get the type 3 endonucleases. Do you mind explaining it again?
Oh, okay. Type 3. Let's go back to this slide now. Okay, so type 3 are enzymes that recognize two separate non-palindromic sequences that are inversely oriented and cut DNA about 20 to 10. So this enzyme type 3 over here, it is kind of like type 1, like it recognize a site. Dia mungkin, yeah, can I draw or not? Cannot draw. My pen. Okay. Now, it is, if for example, you have a stretch of DNA, this type 3 enzyme, it binds right here. Okay, but when it cuts, it will cut like something like 20 to 30 base pair downstream, maybe over here. They will say, hey, dia bind kat sini, how is it going to cut so far away? Well, DNA has, these enzymes has the method of folding your DNA. Because your DNA are just linear punya shape lah kan. It can actually pull it up over here and then cut it right here. Okay, you have to remember, in cell, your DNA is not going to lay out flat, linear like this. It is going to be intertwined or lying flat, overlapping each other. Okay, so when it cuts, it is going to recognize another sequence downstream of it, but the recognition is very specific, just like your ZFN, just like your talent. Whenever they recognize, because there is another region over here, we call it DNA binding domain. Okay, now inside these nucleases, remember, in all nucleases, we have to have DNA binding domain, meaning to say they have to recognize, and then this is the cutting region. Okay, example lah. Okay, dia mungkin terbalik atau macam mana, but one has to be recognizing, the other one has to be the cutting. Okay, so that's why in protein, very often we look at motif, we look at domain. Domain basically has to be attached to a certain function. Over here, this region may be DNA binding domain. Dia bind kat sini. They recognize this sequence over here. They recognize. Bila dia cut tu, dia tidak mau cut ni, dia akan mau cut tempat lain. Okay, like your just like your CRISPR Cas just now. when it recognizes a sequence that you introduce, okay, it has to recognize this PAM sequence. Okay, PAM sequence, when it recognizes, or only then I will cut this region. Kalau tidak, PAM tidak appear, dah maulah saya mau cut. Get what I mean or not? So this is type 3 punya endonucleases. Type 1 punya, they cut at random sites. Meaning they bind here, 1,000 later, only it's simply cut without recognizing where is it actually cutting. Just simply cut. Okay? Boleh? Did I answer your question? Or?
Yeah, doctor.
Okay.
Thank you.
Okay. Any other question from you guys? I'm not confusing you guys, right? Semua dah diam sudah. Dah pergi lunch sudah kan?
No, sir. No.
Okay. You guys go ahead. I need
Actually,
to...
so I have a question, but it's
Oh.
not about the lecture. The lecture is clear. It's about our service learning.
Yeah.
So, our topic that we kind of choose is like breeding and care methods for animal, but in like the purpose of that farm is for meat processing. So is that topic okay?
Can. No worries. The processes, I don't care. I want to know how do they actually what do you call it? How do they actually end up with the product? Yeah. How do they carry out their service lah?
All right. So
Yeah.
let's like we target like for another state to have like a much more like less torturous way to animals, but then we go to an industry that is like more not animal friendly. Is like that comparison is fine.
What? Industry that is not animal friendly. What do you mean?
I mean, like how to say, like some farms they are like there's a lot of force feeding and all of that.
Oh, okay. Force feeding. If you can find one, but if you find one, you dare to report it. It is like a very controversial thing later on lah. And then they are not going to be honest with you as well, unless this manager over there is like, oh yeah lah, we do force feeding, mati kita bagi dia makan saja. Then that's another story already lah. And then you get it recorded. No people will say that in front of cameras or during the interview by the students some more. Mati lah. During coffee talk already. Yes.
It's like okay for two groups to go like to similar breeding and care methods, but then like different farms. It's okay, right?
Yes. Yep. And then you can later on when you write, when you do your presentation, you can get some points from them or some pictures from them, and then you can do a comparison between both groups.
All right. Okay, sir. Thank you.
Okay. Any other questions? If not, we end our class today right here. Okay. Thank you so much for coming today. I'm going to just put my the attendance up again. Anyone of you still want to take your attendance, please take it. Otherwise, you just leave your matrix number, your group number in the chat box. I'm going to help you do it manually if you are using phone.
Doctor, I have a question.
Yep.
For the service learning, right? When we go to visit the place that we want to like learn about, we have to prepare a report so that we can prepare a presentation, right? Or we don't need any report?
No need report. After your visitation, after you have carried out your survey, anything, no need to have report already, because all of the information that you have found, you're going to put it inside your slides.
Okay, so it's directly into slides and presentation. So there's no report. Okay, thank you, Doctor.
When you present that time, you are going to show us lots of figures, tables, the findings. Of course, you have to analyze it a bit. Don't give me raw data, your upper survey outcome. There has been seniors who just directly give me raw data and then ask us to understand during presentation. It's a headache lah. Of course, marks will be deducted lah. So you have to process it, compile the data, the analysis, and then put it into charts or tables or figures, present it to us. Okay, there's no final report, just proposal and then presentation. Yeah.
Okay, Doctor. Thank you so much.
Okay.
Doctor.
Yeah.
If you do like a physical interview, right? Do we have to like
record a video or just like take notes?
If that fella is comfortable with recording a video, then you can keep it lah. You can, this particular video recording, you can keep it and go back to check on the points again. Sometimes you miss certain points. You can re-listen, try to re-understand what he's actually trying to say. Okay, if he allows it. If he doesn't allow it, he said you can only take physical notes, then you might as well take notes lah. If he doesn't mention anything, you just take lah. Some people consider their info are all trade secrets. They do not want to share with anyone. So they're very sensitive into their pictures being taken, their photos being taken, and the recordings being taken. So you have to inquire about it first. These are not like common sense lah, but industrial manners lah. We call it manners lah. Certain manners. When you go to see someone, you will know how to carry yourself. That's very important. Okay.
All right. Thank you, doctor.
Okay. Okay. If nothing else, thank you. 125. Yeah. I'll see you guys. You can leave now.
Thank you, doctor.
Thank
Thank
you.
you, doctor.
Bye. I'll see you
Thank you,
guys.
Thank you, doctor. Have a good
Bye.
day.
Thank you. Yeah.
Thank you, doctor. Take care.
Okay.
Bye-bye.
I'll see you. Bye.
Thank you, doctor.
Thanks.
Thank you, doctor.
Thanks. Bye.